Hello everyone, my name is Moo Sun Hong and I'm a researcher from Professor Braatz group at MIT Department of Chemical Engineering. Thank you for providing me this chance to present about my research at LabRoots Bioprocessing 2021. Today I'll present about model-based control for continuous round activation of biopharmaceuticals. To begin with brief introduction of biopharmaceuticals, they are products derived from biological organisms for treating or preventing diseases. The global sales of biopharmaceuticals have continuously increased for many years, and they are projected to reach about $500 billion by 2026. Percentage of biopharmaceuticals in the drug pipeline have also continuously increased, resulting in hundreds of approved products on the market and over 8,000 medicines in clinical development. The figure in this slide shows a process flow diagram for a typical platform used for producing monoclonal antibodies, which are the highest selling class of biopharmaceuticals. The traditional biopharmaceutical manufacturing consists of a similar sequence of batch unit operations that are divided into upstream and downstream. The upstream unit operations include cell culture with a series of batch bioreactors for protein expression and harvest steps for removing cells and cell debris, which are the centrifugation and filtration in this case. The downstream consists of purification with multiple steps of chromatography and filtration and diafiltration. There are several trends in biopharmaceutical manufacturing providing important rules for process system engineering. First, the growth of biologics increased interest in applying process analytical technology, PAT. PAT is a system of analyzing manufacturing through online measurements of critical quality attributes to ensure the final product quality. Online measurements provide much more data on the multivariable interactions and dynamics. Depending on the process understanding, these data have been used to construct first principles and database models for each of the unit operations. Another trend is a transition of many processes from batch to continuous operation. This is for reducing manufacturing costs and increasing productivity and quality. However, the continuous manufacturing processes also provide new process control problems. Continuous processes have to handle the propagation of impurities and other disturbances caused by the tight integration of continuous unit operations. Another area of interest is the invention of new designs for protein separations. Currently, the dominant method is PEG bed chromatography due to its high resolution, but this method is costly for high-dose pharmaceuticals. So non-chromatography separation methods are becoming more attractive as modern bioreactors are achieving higher product titers. And crystallization is one of the more promising proposed technologies. This will be a major shift in biopharmaceutical manufacturing that will involve new processes to be controlled. These trends ultimately lead biomanufacturing to digital manufacturing. Digital manufacturing is an integrated approach to manufacturing that is centered around the computer system. And again, digital manufacturing is enhanced by using modern systems engineering tools. First, modeling and simulation provides increased mechanistic fidelity and better empirical approaches and can be applied real-time for automated decision-making. Process optimization can be also applied real-time for drug and patient-specific manufacturing. And high-fidelity model-based optimization can reduce experimental costs. Systems engineering also provides tools for dealing with process uncertainty and model-based optimal control. These result in fully automated systems, minimizing errors caused by the human operators. Moreover, synergy between these tools enhances impact to the digital manufacturing. The figure on this slide shows the knowledge pyramid for model-based decision-making. Higher layers in the pyramid correspond to greater knowledge, more informed and better reliability of decision-making, and lower requirements for regulatory oversight. Because higher layers in the pyramid rely more on knowledge and expertise, they are more generalizable and extrapolatable. This makes these models applicable for optimization and predictive scaling. On the other hand, lower layers in the pyramid rely more on data, and they are more specific and limited to interpolation. So these models have limited predictive power beyond the training set. My research applies modern systems engineering tools to develop advanced biomanufacturing systems. For upstream, I derive mechanistic mathematical models and optimal control methods for multiple bioreactor configurations, including microreactor systems and stir-tank bioreactors. And for downstream, I designed and implemented laboratory systems for protein crystallization and continuous round activation. These systems were optimally designed and controlled based on mechanistic mathematical models. In addition, I'm combining these experimentally validated unit operations into a plant-wide dynamic model. This integrated plant-wide model would be able to map the raw materials and operations to the product quality attributes anywhere in the system. In the interest of time, for today's talk, I'll be presenting the work on the continuous round activation system. As I have already mentioned before, the continuous manufacturing of biologics has been of interest to both academia and industry. This is because of the many advantages, such as reduced cost and increased flexibility, ease of standardization and scale-up, improvement in product quality. However, the complexity of these processes also bring control challenges and opportunities. As you can see from the typical process flow diagram, biologics derived from mammalian sources have to undergo two orthogonal virus removal processes. This is to remove the advantageous viruses that may be present in the master cell bank. And one common processing step is a batch low pH hold to inactivate the enveloped viruses. There have been several attempts to adapt this technology to continuous processing, which includes cyclic batch operation, continuous flow tubular reactors, and continuous flow column-based reactors. However, none of these continuous flow reactors directly address the control challenges with operating the system. For the system, critical process parameters are the operating pH and the residence time distribution. These determine the viral activation and also the impacts to product quality from over-incubation or excessive pH adjustment. To address these control challenges, we have constructed a low-cost, column-based continuous viral activation system with off-the-shelf components. For operating pH, we have implemented a fast and accurate model-based feedback control to enable rapid startup and effective disturbance rejection. For residence time distribution, it was estimated periodically during operation through inverse tracer experiments. Then this was used to estimate minimum residence time, which in turn was used to adjust free flow rate. To look into the constructed system in more detail, the figure in this slide shows the process flow diagram of the lab-scale continuous round activation system. First, to bring the fluid to a low pH state, a multi-channel peristaltic pump was used to pump input solution and acid to a mixing unit and inline pH electrode. The mixed stream then flowed into a column packed with inert glass to incubate low pH solution for a sufficient time for round activation. An injection system was used to periodically inject UV transparent solution into the flow stream. UV observance sensors were placed at the column inlet and outlet to measure the column transit. The pH of the fluid leaving the column was raised by mixing with base using a second mixing unit and pH electrode. A programmable logic controller was used for low-level control of the system. Measurement data were received from the pH and UV sensors, and the multi-channel peristaltic pump was connected for control actuation. The programmable logic controller was also connected to computer for data collection, human-machine interface, and online parameter estimation. To start with, modeling and control of pH, fast and accurate control is necessary for rapid inactivation while minimizing impact to product quality. For this, first principles pH model was developed with reaction-invariants, which are total concentration of acid-base species and total charge of remaining species. The basis of the pH model is from the acid-base equilibria and total charge balance with reaction invariance and acid dissociation constants. With the developed pH model, PI controller was implemented based on the reaction invariants, which are now governed by the linear dynamics. This accounts for the nonlinearities in directly controlling pH, which is what we see from the typical buffer titration curves. The uncertainties that may exist in the pH model were handled by maximum a posteriori MAP estimation. This updates primary values based on online measurement data. MAP estimation was formulated as a constraint minimization problem at each time point. It takes into account prior information of parameters and current data to provide improved estimates of the parameters. Estimated model parameters were acid dissociation constants and also this term alpha. Term alpha was used as a multiplicative factor for correcting reaction invariance to deal with flow bias in the feces. Covariance of posterior distribution was also updated each time point with the sensitivities of pH with respect to the estimated parameters. This MAP estimation addresses uncertainties due to natural or unexpected variation, such as changing product and buffer concentration during the operation. So this improves the performance and robustness of the reaction-invariant pH controller. Now let's move on to the low pH hold. Column-based inactivation of reactors have to be designed to simultaneously balance two competing effects of desired throughput and residence time distribution, low pressure drop, and small dispersion. The design variables that we have for these objectives are column diameter and length, particle size, and superficial velocity. In a column-based design, throughput is scalable to the diameter of the column, so this variable is not independently adjustable. Having a fixed average residence time also relates column length and superficial velocity, so we can consider column length to be fixed as well. Pressure drop from Kalman-Coushain equation can be reduced to the given formulation. In a similar way, the standard deviation of a dispersed peak can be characterized as given formulation from the correlation of the dispersion coefficient. And we can see in both relationships that the term superficial velocity over particle size only appears together in the formulations. So we can conclude there is no way to optimize a choice of particle size or superficial velocity to simultaneously reduce pressure drop and dispersion. But in the interest of system compactness, a small particle size and superficial velocity are preferred for the system design. Quantitative understanding of low pH hold is important to balance two competing effects of Brown activation and degrading product quality. This comes in the form of residence time distribution, which describes the time that a fluid element spends within the system by probability distribution function. The column was modeled as plug flow reactor with axial dispersion in series with a continuously stirred tank reactor, with the respective residence time distributions shown in the given formulations. Plug flow reactor with axial dispersion describes the cylindrical chemical reactor with continuous flow, and continuously stirred tank reactor describes any non-ideality that might be present in the actual column. Then the concentration at the outlet of the column can be calculated through the convolution of the concentration at the inlet and the residence time distribution of the column. And again, residence time distribution of the column can be calculated through the convolution of already modeled residence time distribution of plug flow reactor with axial dispersion and continuously stirred tank reactor. The cumulative distribution function can be also simply computed by integrating the residence time distribution over time. And this allows us to define the minimum residence time as a given formulation. Term eta here is a small value corresponding to the fraction of material leaving the column before the minimum residence time. This residence time distribution is affected by several variables such as reactor design, geometry, fouling, and input flow rates. And these variables can be classified into three different types. Design variables are those that can be controlled offline, like reactor design and geometry. Disturbances are those that may vary during operation but cannot be effectively controlled, like fouling. And manipulated variables are those that can be controlled online, like input flow rates. While design variables have been explored quite extensively by previous work, disturbance rejection has not been discussed directly. Actually, disturbance rejection is even more important, as these continuous reactors will be operated for extended period of time in pilot or commercial scale. Because residence time distribution is infinite dimensional, it is impossible to completely reject the disturbance with scalar manipulated variables. So this has to be done on a scalarization of residence time distribution. One of the scalarization is logarithmic reduction value approach, where a nonlinear model is used to relate residence time distribution to scalar logarithmic reduction value. This approach directly addresses the critical quality attributes, but it relies on an accurate inactivation model. However, inactivation kinetics vary from virus to virus, and they are uncertain during the process development stage. So we avoided this uncertainty by utilizing the minimum residence time approach. The refraction used for defining minimum residence time varies from 0.05 to 10 to the minus 5 in the literature, and we used 0.05 fraction in this work. At the same time, I would like to mention that different fraction, or even logarithmic reduction value approach, can be trivially implemented with the information of inactivation kinetics. For controlling minimum residence time, three components are needed. First, means of measuring residence time distribution and minimum residence time. Second, feedback control algorithm for adjusting flow rate in response to minimum residence time measurement. And lastly, means of actuating the flow rate. First, measuring minimum residence time directly using tracer experiments is impossible due to small fraction value and inline sensor noise. To overcome this lack of observability, residence time distribution was estimated as a parametric distribution, which then can be used to determine minimum residence time. Using the Beard-Lambert law, the absorbance is directly proportional to the concentration. So the parameters for residence time distribution model can be calculated through the given sum of squares minimization problem. Sufficient variability is necessary in the inlet absorbance for the accurate parameter estimation. This can be achieved through the normal process variation in the system, but also safely through the periodic injection of protein-free buffer, which has negligible UV absorbance. Given the minimum residence time estimation, a feedback control algorithm was developed as given formulation. Because peristaltic pump flow rate are almost linear to pump rotational speed, the system can attain the set point within a single step after the residence time distribution measurement. Any nonlinearities or disturbances, such as tubing stretch, are also addressed through this feedback control. For experimental validation of the described system, we have conducted experiments to demonstrate the controller performance. These experiments were planned to ensure that the solution pH is correctly low to the target pH for vial activation, solution residence time is accurately and precisely controlled, and the solution pH is correctly raised back to the target pH for further downstream unit operations. The first experiment was planned to demonstrate that the system is capable of tracking pH and minimum residence time set points. Second experiment was planned to demonstrate that the system is capable of suppressing pH disturbances. This was done by changing to a different buffer for pH control without updating the initial parameter values. Last experiment was planned to demonstrate that the system is capable of suppressing resonance time disturbances. This was done by using a bypass line to suddenly decrease the active volume in the system. For these controller validation experiments, 0.1 molar sodium hydroxide was used as the test input solution, with 1% acetone used as a tracer for the absorbance measurements. 1 molar phosphoric acid was used for the acid stream, and 1 molar sodium hydroxide was used for the base stream. The ionized water was used as a low absorbance injection solution for the resonance time distribution estimation. Plots on this slide show the experimental results for tracking pH setpoints. Plot on the top shows flow rate determined by the pH controller, and plot on the bottom shows pH measurements with set points shown as black dashed lines. During the experiment, the set point for first mixing unit was changed from 3 to 4 at 60 minutes and reverted to 3 at 120 minutes. pH set point for second mixing unit was also changed from 7 to 8 at 180 minutes and reverted to 7 at 240 minutes. We can see from these plots that the set point tracking was achieved with fast and accurate response and no instability. Plots on this slide show the experimental results for tracking minimum resonance time set points. Plots from the top show flow rate determined by the minimum resonance time controller, absorbance measurements at the inlet and outlet of the column, and estimated minimum resonance time with set points shown as black dashed lines. During the experiment, minimum resonance time set point was changed from 240 seconds to 300 seconds at 180 minutes and reverted to 240 seconds at 360 minutes. First, we can see from the second plot that the resonance time distribution model with estimated parameters well describes the concentration at the outlet. Also, we can see from the bottom plot that the set point tracking was achieved within a single step after the resonance time distribution estimation. Moving on to the next validation experiment, disturbance rejection was tested for pH control by changing the buffer concentration of the acid stream from phosphoric acid to citric acid. This was done with the model still being initialized with the acid dissociation constants of phosphoric acid. So the model is initially not aware of the buffer change in the acid stream. Plots on this slide show the experimental results for the disturbance rejection of pH controller. From the top, plots show flow rate determined by the pH controller, pH measurements with set points shown as black dashed lines, and the estimated primary values of acid dissociation constants with true values for citric acid shown as black dashed lines. We can see from the second plot that the set point tracking was again achieved with fast and accurate response and no instability, even with the introduced disturbance. We can also see from the bottom plot that the MAP estimation algorithm correctly estimated the acid dissociation constants of citric acid purely from the pH measurements in the first few minutes. For the last validation experiment, disturbance rejection was tested for the minimum resonance time control by adding an additional length of tubing in series with the column. While the system is in normal operation, tube clamps were used to remove this additional length of tubing in the flow path as shown in this process flow diagram. This effectively led to suddenly decreasing the active volume through the column and therefore suddenly decreasing the resonance time in the system. The plots in this slide show the experimental results for disturbance rejection of minimum resonance time controller. Red vertical line shows the time and the length of the tube was decreased with the tube clamps. We can see from the second plot that the model correctly predicted the output of each water injection even after when the sudden disturbance was introduced into the system. And we can see from the bottom plot that the minimum resonance time measurement correctly identified the sudden change in the system resonance time. Then the controller adjusted the pump speed accordingly and restored the original resonance time set point again in a single step. To conclude, in this work, we constructed a low-cost column-based continuous file activation system with off-the-shelf components. and controller validation experiments demonstrated that pH and minimum resonance times setpoint tracking and also disturbance rejection are achieved with fast and accurate response and no instability. Overall, this work provided tools for the design and operation of continuous bioactivation systems in service of increasing productivity, improving product quality, and enhancing patient safety. With that, I would like to acknowledge my coworkers listed here and also the funding source, U.S. Food and Drug Administration. And thank you all for your attention.